Thank you very much. I welcome you and I will start my presentation. So you should see now.
Okay. Yes, we see.
Okay, so as Jean-Christophe said, it's now the identification of the subunits. So what are the immunogenic parts of the 3 viruses we selected as models? And this is for Schmallenberg, given by me. Jerome will talk about Rift Valley fever, and Bart will talk about the MERS coronavirus. If we look at these 3 targets again, you see, meanwhile, there is structures known, and there is special proteins we concentrated on, but you will see this in the next slides for Schmallenberg virus, Rift Valley fever, and MERS coronavirus. And this is very similar parts and the same more or less proteins you have heard in the session about the antibodies, because you also see there's a lot of exchange of knowledge between the 2 parts, and we can use, for example, the structure for both analysis. What are the best antibodies and what is the best antigen to induce antibodies? So this is highly interconnected. So I would start with the ZAPI approach applied to the Schmallenberg virus model, which was mainly done at the FLI. So if we go to the Schmallenberg virus and the key immunogen selection, the first question was, which protein is the right one? And from autobunya viruses, even if it's a completely new virus as Schmallenberg virus, you have some relatives. And there it was clear the envelope proteins, Qc, Qn, are the first candidates, which are shown here. And if we look at the M segment, which is coding these ones, you can see there is GN, in the middle is an NSM, a non-structural protein, which connects GN to GC. And from other viruses, it was known that both might play a role, but maybe GC is more important. So we could concentrate on this one first. And the question was how to develop systems to find out what is the most immunogenic part. Is it the whole protein? Do you need always the whole virus, which would be not good for alternative systems? Or can we use only small parts or even peptides? And therefore, we did a lot of in silico analysis and in vitro steps. And the first one was looking at this and taking the GC out of it and all the others, so the whole segment, and divide it in logical parts. Express these parts and see what is reacting with anti-sera, for example, or monoclonal antibodies. And it was on one hand very clear that the GC is the main target. GN does not play any role. So we could concentrate it on this one. And if we look at the reactivity, it was also clear that we have here a very small part from this overall segment, which we call the GC amino, the amino terminal part, which more or less makes all the reactivity in monoclonals and most of the reactivity in sera. So we could define by this approach to put it down to a smaller domain, which is only a part of the overall segment and only part of the immunogenic GC. So the main immunogen is GC, and the main domain we called at this time point the GC amino. You will later hear that this has now a new name coming from the normal, more or less normal nomenclature of most of these viruses. In the next step, and this first work was mainly done by Kleider Roman Sosa as a postdoc, looking at all these dissected parts of the GC. And he also could show that there is in this GC amino, there is additional, he called it clusters, where the monoclonals bind and which are connected, for example, by cysteine bridging and glycosylation sites. So we also could say glycosylation is important, and this 3D structure at the end will be a big part. And in the project, and I think this is a really high achievement, high-level achievement, the group of Felix Ray and Pasteur and Jan Hellert, he was the person doing this experiment. They succeeded in the crystal structure of this domain, and this is now, so from Qc amino domain, it's now called the Qc head domain. And this head domain is the most immunogenic part, and there is 2 stalk stub domains building 1 stalk domain. So in the project, we came from this very rough prediction, Qc might be important, and parts of Qc down to the structure, showing that this is important parts and how they are in the 3-dimensional structure, folded, etc. And this amino terminal part, this head, is on the spike head of the glycoprotein C. And we therefore concentrate in the next step here on this short head alone, and the head stabilized by the stalk. We called it now Qc head and Qc head plus stalk. And we did additional PEP scan and in silico predictions, finding in this Qc head 2 main peptides, and one, which is the one here from 694 on, which we could show that it is reacting with sera, for example, and is very reactive. So the main goal in the ZAPI project to define immunogenic key domains was possible in this effort. And I will show you later that this is also something we can transfer to other related viruses. Is this now really immunogenic enough for vaccine? So we started with a lethal animal model, the IFNAR mouse model, and the Qc head could completely protect from clinical infections, or no animal died, but the Qc head stalk even gave sterile protection. So this was a step further. So the stabilization of the immunogenic part is obviously important. If we look only at the Qc head, we did also target experiments with a subunit alone, and there was sterile immunity in 3 or 4 animals, but also a very good antibody response. So the head alone is the immunogenic part. Qc head plus stalk gives a superior reactivity, obviously. We wanted further to improve that, because one part in this project was also, can we use these key immunogens, which are now smaller, can we use them in improved systems? And you have heard of these superglue, the SpyTag spycatcher, and we concentrated on multimeric scaffolds, the lumacine synthase, and we compared that to the subunit alone. The subunit connected with the superglue on the lumacine particle, and also the peptide alone. So is a peptide on a particle enough? And we will later on hear much more, but again, here in this nice animal model, we compared the subunit and the particles with the Qc head and the stalk, and you could see immediately there is complete protection, and even no body weight goes down. And it's obviously enough to have 1 immunization if you use the head domain with the stalk. So again, also here, it shows it's a very high immunogenic region, and together with a particle, so further improving it, adjuvants, particle, and the right domain, you can even get full protection after 1 shot in this mouse model. So I think this shows very nicely that we were on the right path. And there's a lot of these viruses. So if you look at Smallenberg, all the relatives are here on the map. And Oropushi is a virus which is also zoonotic. We have Shuni, which is spreading further. It's in Israel now. And there is newer results also showing some zoonotic potential. And what we could say at the moment is we can really transfer this and can use the same domains from the other viruses to get the same level of reactivity. So we did this for Akebani virus, Oropushi, and Shuni virus. And I think this shows also that the transfer is possible. So all this work speeds up everything for the other viruses. So if I summarize the Schmallenberg part, we have Qc as the main protein for immunogenicity. We have the Qc head domain where more or less all the neutralization is focusing on. And we can enhance this, what we learned from the structure, from a stabilizing stalk domain. And then we get in our first experiments an enhanced sterile immunity, and we could also transfer this to related orthobunia viruses. So I think we could fulfill this ZAPI goal we had here for the model virus Schmallenberg. And with that, I hand over to Sharon, showing the more or less similar approach for the Rift Valley fever virus model. Sharon, the floor is yours.
Yes, thank you so much. Okay, so Martin, since you have the consoles, I will ask you to move also through some animations in the presentation, if that's okay.
Yes.
Okay, I will be presenting the application to Rift Valley fever virus, and I'm Paul Wiggerscheur is mentioned here as well. He was in control of the or taking the lead in the laboratory work, and he will explain later today about the validation of our results. So this picture shows just how beautiful Rift Valley fever looks like, and if we want to identify the domains that are targeted by neutralizing antibodies, obviously it would be good to have a bit of a closer look. Could you press the next, please? Yes. Okay. So this is the schematic overview of what the particle looks like. So it contains, like Schmallenberg, 3 genome segments, of which the medium genome segment encodes the 2 structural glycoproteins, GN and GC. Next, please. So before ZAPI started, we already knew that GN is the primary target of neutralizing antibodies. This was already described in the 90s, and we were studying this this protein ourselves as well in my lab, also before ZAPI. And yes, next please. And so what we did within ZAPI is we want to have a closer look in these domains, because as for Schmallenberg, we were aiming to produce as small as possible subdomain, because in that way we could manufacture the protein in very high quantities for emergency application. And for this, we needed to find out what the key immunogens are within this GN protein. And so for this, we used a PEP scan analysis, where we use linear peptides, but also peptides with loop structures to find linear epitopes, but also discontinuous epitopes. And we used sheep sera, so from sheep that have been infected by the virus and that have convalescent sera. So if sheep survive, they develop very good neutralizing antibodies. And with using those sera, we were able to identify 6 continuous epitopes and 3 discontinuous epitopes in the GN actor domain. And also we've used well-described classical antibodies with high neutralizing capability, and also used those to validate our approach. Next, please. So this picture shows the schematic overview of the polyprotein that's produced from the M genome segment, and here you can see the GN and GC proteins, and these are post-translationally modified and cleaved into the individual proteins. Next, please. So what we did, we looked at, together with a lot of help from our ZAPI partners in Institut Pasteur, we looked at the structure of this, looking at taking into account the cysteine positions and also hydrophobicity plots, etc., and looked at what part of the protein could individually be expressed. Obviously, these proteins, these subdomains, had to be folded correctly to elicit the right immune response. Next, please. So what we did, we selected these different subdomains, and as you can see here on the SDS page, some of these subdomains were expressed to very high levels, others were not expressed at all. And you can already see that a number of these proteins were primary candidates. Next, please. And then if we looked at the recognition of antibodies, neutralizing antibodies to these domains, next, please. You see that both the yield of one of these constructs, so domain 7, was the highest, and also had the best antibody recognition. So this is still quite, it's a bit of a bigger protein than the one developed for Schmallenberg, but still, as you can see, in the yields were somewhat better than the whole actor domain. Next, please. Okay, so within the ZAPI, this was not done within the ZAPI project, but when the project was running, the crystal structure of the GN actor domain was published, and it looks like this, and also shown this morning already. And it was really very nice that we actually predicted this domain 7 actually corresponds very nicely to this structure. Next, please. Next. Yeah, so this is also what we call the head domain of Rift Valley fever GN. Next. And this picture I already shown this morning, and this shows how this ectodomain is positioned in a pentrameric in this typical ring-like structure on the Rift Valley fever variant. And what as I showed this morning, we've shown with combining these VHHs targeting the yellow site and the blue site depicted here, that you get optimal protection by these neutralizing antibodies, and thereby we also validated that this domain. From both looking at the neutralizing antibody perspective as well as from the antigenic perspective is really the key immunogen of Rift Valley fever virus. Next slide. Yeah, so what we did on the left, you see here how we produce the antigen. So this was produced with an N-terminal spike catcher using the baculovirus expression system, and we've used baculovirus because we do need at least one glycan on this subdomain for correct folding. And on the right, you see the multimeric particles, scaffold particles, where we used 3 different scaffold particles, actually the lumazine synthase, the aldolase, and the E2. And these particles are of similar size, and they also have the same number of subunits, but the production, of course, of these particles could be very different. So we valued it a lot to look at more than one particle. And we fused the spike tag to the C-terminus of these proteins, produced them in E. coli to very high levels, and then assembled these two together, yielding the particle that you see in the center, expressing the ectodomain of GN. The next slide, please. So what you see here on this gel is to the left, you see the GN head with and without spike catcher. Then on the same gel, you see the lumazine synthase with and the E2 and the aldolase with a spike tag. And in the right gel, it's a little bit complicated, but if you look carefully, you see that if you put the GN head without spike catcher together with the particles, you see the individual components in the gel. But if you have the spike tag and the spike catcher, you see a combination resulting in these coupling of the particle and the subunit. And then if you look on the right, you see on the top level of top pictures, you see the particles with the spike tag without the subunit coupled, and at the bottom you see the particles with the subunit coupled. And as you look carefully, you see that there's more electron density around these particles, suggesting that indeed these are the particles with the decorated subunit attached. Next slide, please. Okay, so we did a first validation in the mouse model. So these are BOB-C mice, where we used the lumazine synthase particle with the GN head domain. We compared coupled and uncoupled antigen to investigate if the coupling really adds to the immunogenicity and the efficacy. We used a 2-time vaccination regimen and a challenge after 2 weeks. And as you see in the body weights and also the mortality curves, you can see that we got partial protection if we only when we coupled the subunit to the particle. So with that, we've shown that the coupling really adds to the protective efficacy and the immunogenicity. However, we also included groups where we used an adjuvant, and this is Stimune, water in oil adjuvant, and we can clearly see that the Stimune has a big contribution, and only with the Stimune you get complete protection from mortality. But nevertheless, this still looks very promising because the mice are very susceptible to Rift Valley fever virus, so it's quite difficult to protect them from lethal disease. And if you look at the bottom left, you see the neutralization titers, and this corresponds to the efficacy we see in the survival curve. So with Stimune, 2 vaccinations, you get very high levels of neutralizing antibodies that correlate with protection. And next slide, please. So concluding from this is that at least in the mouse model at this time, we've shown that the GN head subunit is a potent immunogen, and that if it's coupled to the nanoparticle, you get good neutralizing antibody responses, and it reduces or even prevents morbidity and mortality in the mouse model. And as I said, Paul will present later today the validation in the sheep model. Thank you.
Sure. Thank you very much. So I think it shows nicely the two different proteins in different Bunya viruses, but a very similar, very successful approach also with these particles. We will go on now with Bart, talking about the ZAPI approach applied to the MERS-CoV model. Bart, the floor is yours.
Yeah, thank you, Mark. So it's clearly important to state that Rift and Schmallenberg were clearly the two targets for further manufacturing and development of the vaccines, but I think we also want to show proof of principle for MERS-CoV that also in this case, the selection of a domain and then coupled to a particle would work and enhance immunogenicity and protection in this model system. So this slide you already have seen this morning, as presented by Bear and John, showing the spike protein with the S1 domain and the S1B domain. The A and the B domain, the A domain binding to sialic acids, and the B domain, the receptor binding domain that is critical in the interaction with DPP4, the receptor, whereas the S2 region is involved in fusion and subsequent uptake of the particle. So next slide. So the approach we took is to first look at neutralizing antibodies induced by a vaccine that protects, and in this case, in the dromedary camel, that is the intermediate host. So an MVA expressing the full-length spike was used, and these animals were challenged. You can see on the left that only the control showed nasal discharge, but the MVAS vaccinated animals were protected. Also, with respect to excretion of virus in the closed bars, you see infectious virus from the nose, and that is limited in the open bars, the animals that were vaccinated with the MVAS. And on the right, you see also that next, the in situ hybridization shows limited expression of the RNA in the nose of the vaccinated animals. So these were interesting animals to follow up on the antibody response. Next slide. And actually, this is also what Martin indicated. So in line with experiments done to look at neutralizing antibodies to be used for therapy, in this case, we used the antibodies that were screened from the bone marrow. So that is the VHH that were put on an FC for analysis of neutralizing capacity by direct cloning from bone marrow cells. We analyzed all those that neutralized MERS-CoV. And interestingly, in the middle panel, you can see that when these were screened for reactivity to S1 and RBD, all the neutralizing antibodies actually reactive with S1, but also to the RBD. And these are very potent neutralizing VHAs, and as shown on the right, in that in the nanomolar range, I should say in the picomolar range, they're already neutralized. So by this, we pinpointed to the S1 region as important for the induction of neutralizing antibodies. Next slide. And the first experiment done, and that is in collaboration with the group of Qcsa in Barcelona, we set up a transmission model in llamas, and that's shown in the upper part of the figure, so that 3 animals were inoculated. A day later, 5 animals were put next to in the same room to see whether the virus can be transmitted. And on the right, you can see in red that in the recipient animals, so the 5 animals that initially were naive, infectious virus can be detected. In contrast, if you do a vaccination with S1, recombinant S1, with an adjuvant through the intramuscular route, and that's shown in the lower panel. So now the 5 animals are vaccinated, and in this case, as shown on the right, no infectious virus could be detected in the recipient animals, indicating that the S1 can induce protective immunity, prohibiting efficient replication in the recipient animals. Next slide. So to further follow this up is, as I said, these antibodies also target the RBD domain within S1. So in line with the earlier experiments shown for the other viruses, we now use the RBD, and that is work done by Ben and John Bosch, to couple the RBD using the spike catcher system to the limousine synthase, and it's shown here. And this was then tested together with our clinics in a rabbit model to see whether these particles are immunogenic and can protect against microscopic challenge. Next slide. So that's shown here. On the left, you can see the antibody titers, so indicating that coupling enhanced antibody titers. So there's a log scale, so these are 10-fold higher antibody levels that are capable of neutralizing the virus. It's shown in the middle. And here also you can see that on the right in the boxed plot is the animals that were immunized with the coupled RBD. It clearly showed high levels of neutralizing antibodies. And on the right, you can see that infectious virus shed from the nose is absent in the animals that were vaccinated with the RBD limousine synthase. So clearly indicating a beneficial effect of the coupling. Next slide. So this more or less closes, and we're still analyzing this in a llama model. But for sure, this was of interest, not only for us, but also many groups. And there are 2 slides to show that we're working on a C1 express RBD together with the ADIC to see whether we can move the preclinical experiments here with this setup for SARS-CoV-2 into a phase 1 trial. And of course, there are many other initiatives at that level, and one is, for example, also the SARS-CoV-2 vaccine using RBD coupled to HBs antigens, which is done in the Serum Institute in India. Next slide. But there are many different initiatives, and for example, on the left you see an article also presenting data that the nanoparticle system is suitable to induce high levels of neutralizing antibodies when using coupling of the RBD, and that's work also done by the group of Neil King. That is followed up also with the full spike protein work done at the EMC collaboration with the group of Neil King. But interestingly, you can also use this approach not only to couple one RBD, and that's shown on the right, but you can in principle develop mosaic nanoparticles that contain different RBDs from different related, for example, SARS-CoV viruses to induce antibody responses that are broadly reacting. And I think there's an interesting development, but also recently published work. Next slide. And with that, I think I close this session and give the word back to the chair.
So thank you very much. I think with here with all the names, I have really to say it was fun to work together on this, and it was for me a real surprise that we were able to find these smaller domains. And I think we can in general learn that around 250 to 400 amino acid domains obviously are the ones giving enough stability, enough folding to get enough immunogenicity. And if I compare it with some of the full inactivated virus vaccines, the small domains, to my feeling, were even better than the whole viruses in most cases. So it makes really sense to put it down to these domains, and it is successfully possible. And for the related viruses, I think also we learned a lot, and it can be used more or less immediately, and not with a lot of new work starting and finding out. Learning from the other viruses will help a lot. And with that, I thank all the people who worked here, and give back to Jean-Christophe. Thank you very much.
Yeah. Thank you, Martin. Yes, very, very good presentation from the 3 speakers. And you have seen that indeed, for me as well, it was surprising to confirm that small subunits could be enough to induce very good protection. Especially when they are displayed on particles. That's something that was developed during the project, not initially. We had some idea to make fusion proteins, but not in this way, because the superglue technology, and we'll discuss more later, appeared during the project, and we took the opportunity to use it immediately for this project. So now I will give the floor to Alexander and Ronen to explain how the initial screening of platforms was made, comparing Baculo, C1, and E. coli, and other systems. You have seen some yields at the bench scale, but when you consider to achieve millions of doses or hundreds of millions of doses, indeed the yields have to be some orders of magnitude greater than what you get at the bench. So Alex, will you present the slides from your end, or do you want me
I
to?
can. Give me 1 second to see if that works, but I think it's easier if you do it.
Okay.
I'm happy to help if you want.
No, it's okay.
Sure.
I can do it here.
Thank you very much.
There you go.
Okay, so hello everybody. As Jean-Christophe said, Alex Briggs. I'm affiliated with BI with the Hanover site, and as you will see, we were tasked with starting the production of some of these materials, and Ronan and I will now talk a little bit about the initial screening. So basically what came from our partners. You just heard about the different antigens, for example, and then how did we take that into our labs. And if I talk about production, I think that's session 2C, so I'll go into more detail a little bit there in terms of scale and so on. Next slide, please. It was just mentioned for the vaccine, the approach was chosen to couple the viral antigens on selected MPSPs. Probably heard about all these advantages, these self-assembling particles, monomers. Some of them are resistant to temperature, which we actually use then during the downstream processing. To make the story short, when we started, we looked at 4 different particles with each spy tag and spy catcher configuration. So initially, together with BI Animal Health in Lyon, actually, we had some screening work done to see which of those are actually producible, let's call it. Next slide, please. And I will just give you an example. It was just talked about mostly about the LS, lumicin synthase particles. So that was one of the initial ones Hanover focused on. And we basically had very screening done with very standard parameters, 2 clones per contracts, different media, different induction temperatures, excuse me, to see which of those combinations are actually producing, which can we take further into a later development. And this was all done in milliliter to liter scale. We did not focus too much yet on the DSP, but next slide, please. The initial results, or the example, as I said, that I chose here, the lumicin synthase, we were able to produce different materials. The spy tag coming from inclusion bodies, for example, as is described on the lower left, with an ion exchange, refolding, and size exclusion chromatography. This material was then actually provided to our partner for an animal study. But we also had, for example, the lumicin synthase spy catcher that was successfully produced at a lower yield, but it was still, we were able to produce it. Next slide, please. So we had a lot of different information from Lyon, from Hanover, and then based on this information and based on the results, the data, we in the team decided to focus on 3 different MPSP configurations. So it was lumicin synthase spy tag, the aldolase spy catcher, and the E2 spy tag. And with those 3 candidates, we went into the next round of screening. Based on the fact that we knew we can produce them, we also wanted to look at the downstream. And we, of course, had the goal to have a relatively easy downstream process, as always from an industry perspective. And the result was basically, if you all take this together, the yields and the yields post in upstream and the yields post downstream, and the easiness of those procedures, we decided to focus, after a lot of discussions, also with Jean-Christophe, of course, focus on aldolase, SpyCatcher, and E2 SpyTech as the two candidates that we will take further into what we call medium scale, which I will talk about in the next session. As a rough start already, let's say, E. coli production, induction, basic induction, and then the downstream comprise of ELISAs, the heat treatment, and the size exclusion chromatography for the aldolase, still to be improved. But this was the start, and for the SpyTech E2, basically we had an ion exchange chromatography developed at this time for the downstream process. Next slide, please. But we did not only have those MPSPs, we also had to look at the viral antigens. And you just heard about the insect cell work that was done at our partners before, either in also baculovirus or in S2 cells. Here, the two candidates that we worked with were the Rift Valley fever virus domain 7 and the Schmallenberg, I think it was called head domain, that were basically provided to us. The information was provided to us, and we then took these two into into the production. Our approach was as followed. We had externally the baculovirus construct being prepared or generated. Still, like with the MPSP, different versions with SpyCatcher and SpyTech, and also, which is not listed up here, further down below, with or without StrepTech for initial purification purposes. Why baculovirus, for example, and not S2 baculovirus? Because of the fact that it's an established industry system. BI has a lot of experience with that since approximately 2006, 2005. And there's a lot of process knowledge I'll talk about a little bit later. Scale up, scale down, it was mentioned by AstraZeneca earlier today. So from an engineering perspective, it's very well defined as well. For Hanover, that meant in this work package, we had to start those cells. We focused on SF, Spodoptera frugipera insect cells, of course. We optimized the cell growth of different cell lines in different cell culture media with the statistical design of experiments in a shake flask system first. And then once we had optimized those conditions for cell growth and viability, we looked at initial antigen production in shake flask with a focus on the Rift Valley candidate. Next slide, please. Once we saw we were actually able to produce, we were also looking into of course, into the production system, which is a classical stirred tank bioreactor, as you can see here. Here, of course, lab version in glass. In production, it would be either stainless steel or single use. As I just mentioned, there are established scale down, scale up parameters or parameters, let's say, criteria. There is a lot of process knowledge. We basically took our 4,000 liter production scale and scaled it down first to the 1 liter and then to the 5 liter system that we have in the lab based on our knowledge and our engineering aspects or parameters. And with the cells we were using, we validated those settings that we calculated to see if that's actually, true, basically, if it's working. Next slide, please. The result was that it was working with cells. So we went ahead with virus infection and protein production. And the results, and this is the conclusion of the screening phase for baculovirus, was that yes, we can produce, but we already saw some problems, some issues at that time that we had to work on. And basically that was twofold. The Rift Valley candidates, basically all of them that we had generated, showed a very significant decreased expression of the antigen with increased seed passage. And if you're familiar with the production, we usually have at least 5 seed passages where the virus needs to be stable for production. So that was a big issue here. And the second one for the Schmallenberg, it was easier to solve. We basically only had to optimize the harvest time because it seemed that the antigen was in the culture liquid or culture broth wasn't very stable. But we ended with the 1 liter scale at this point for the screening purposes. Next slide, please. Especially for the Schmallenberg, we saw these questions. We then said, okay, we have this 1 liter process, but usually we also want to optimize the process. And as before for the shake flask, we applied statistical design of experiments to improve or to optimize the parameters for this production as well in the 1 liter scale, namely the dissolved oxygen content and the gassing strategy. So which gases do you put in to achieve the oxygen level that you actually want? Next slide, please. I think, Ronan, this is for you now for the C1 system.
Okay. Thank you, Alex and Jean-Christophe. I think we heard all about the nice identification of epitopes. And also we heard with Alex the initial way to try to express it in other production platform. But as we know, production platforms should be very important in order to really be able to bring this project into commercial scale. So in the next slides, I will present the C1 expression system and its contribution to the success of the project. C1 is a filamentous fungi named Thermotelomyces heterothallica, that previously was Mycelophthora thermophilia. And this is Dyadic developed this production platform for the production of enzymes to the bioindustrial field. But since 2016, we shifted the target to pharmaceutical-based proteins. Actually, we have started to leverage the technology to pharma as we joined the ZAPI project. Since C1 was a new candidate, we received moderate goals to express of at least one antigen of each of the 3 target viruses. Second, to express stable antigen subunits. And overall, the expression level that we should reach at least 100 milligram per liter. Initially, we could demonstrate that C1 was able to express and produce those 3 antigens. At the beginning, without the spike tag and the spike catcher, in order to couple it to the particle. And we actually expressed the antigen for the MERS RBD, the Schmallenberg virus, and the Rift Valley fever virus. All of them were secreted into the media, but the productivity and the stability at the beginning were compromised. An example of the MERS RBD expression could be seen in the next slide. Here you can see a 24 well plates expression. And as you can see here, different promoters generated different expression level. But still, as you can see here, with the CBH1 promoter, we could really see a very high productivity, which according, at least on this gel, was overexpressed. But altogether, the expression level was not high enough. And also, as I said, the stability was not really enable us to qualify those cell lines for commercial production. So later on, I will demonstrate the results we obtained with the antigens that were coupled to the spike tag or the spike catcher, and with a level that I believe we can convince you that can support a commercial scale. Okay, so thank you. And I will come back to you when we describe the current productivity and status.
Thank you, Ronen. So this will end the 2B session, and I want to thank all the people, technicians, scientists who participated in this part. There are quite many, and especially the partners, because initial work was done, as you heard, at FLI in Martin's team, and at WBVR with Jeroen and Paul especially. We are a little bit in advance, so it's good. So we may have our 10 minutes break now, and I suggest to meet again at 3 p.m. instead of 3:10. Hello, welcome back. I think we can start now session 2C. Alex, Ronen, are you okay to start?
Yes.
Okay, I will drive the slide, and I will start commenting. So this session is on the scale-up of different components of the platforms, and we'll describe how we selected the 2 platforms that are currently used for the MPSP, so the scaffold particles and the subunit. And then we'll have a section on the analytical tools. So as you have understood, the ZAPI vaccine approach is based on 2 component system. The scaffold, MPSP, multimeric protein scaffold particle, which is expressed in E. coli right now, but could be expressed in another system later on. It is fused to one part of a superglue system, here SpyTag as an example, but it's flexible as you will see later. And the subunit that is fused with the other or the counterpart of the SpyTag, which is SpyCatcher, so complement of the superglue system. And the final product is actually the coupling between the 2 components by a simple mixing that we'll describe. And this new technology, bacterial superglue, as it was described earlier by Jeroen, comes from the Oxford University in the laboratory of Mark Howard in the biochemistry unit. And for me, it's a really remarkable molecular biology breakthrough. It's kind of dream, because you can start to play with proteins as I was playing with Legos when I was a kid. And it opens also a lot of possibilities for designing new proteins, new vaccines, a lot of things outside of ZAPI objectives. We have described earlier with Alex that the 2 candidates that were selected for medium and large scale are the 2 combinations, the E2 SpyTag with the Rift Valley domain 7 fused to SpyCatcher, and the aldolase SpyCatcher with a different multimeric scaffold to be coupled with the Schmallenberg subunit fused with SpyTag. So this is to show that we have actually a flexible platform that can be used in different ways. We have only 2 examples here, but probably there are possibilities to develop more in the future. The initial idea was to go with, still pursue the baculovirus system because it's a major industrial system that we used for commercial vaccines made at very large scale. But in parallel, Dyadic, and Ronen will describe that, pursued the refinement of the system in order to increase the yields, especially based on a key advantage, which is the secretion. And just to show the flexibility of the system is that, in fact, either SpyTag or SpyCatcher moiety can be fused at the N-terminus or C-terminus of the scaffold, depending on the scaffold, but typically both ends are free to access, and you have different possibilities to modify and to generate or to design in order to have a better coupling system. Just a few words, and because we would need or maybe a full week to describe what are these MPSPs. In fact, we are just leveraging unique features and biophysical properties of natural proteins, which are self-assembling particles made of proteins, typically with dodecahedron symmetry with 60 monomers. So very like viruses, I would say. Typically, they have a high resistance to temperature, more than 65 degrees Celsius, and some of them can stand boiling because we come from thermophilic bacteria living in volcanoes, geysers, deep sea springs, and using a high stability. And it is likely a very large family of natural protein scaffold, and we know just a few of them so far. So it's also a source of innovation for the future, just if we look into this part. So Alex, the floor is yours now.
Thank you, Jean-Christophe. I would like to continue with the production aspects that we were just talking about a little earlier. You just heard that we had focused on, or we have focused on 2 MPSPs. The first one was the spycatcher aldolase. And I just want to give you a brief overview on how the production process that we developed looks like and where we are at the moment. In terms of upstream, as you can see, the information during the screening was then used to move this from basically shake flask cultures to, in the end, eventually 5-liter scale fermenters, as you can see on the picture here. For us in the lab scale, it is the medium scale, but of course it's not a commercial production scale yet. It's a very robust and easy process. We will go, maybe Jean-Christophe, we can talk about a little bit earlier, but of course the 5-liter scale is not the end. This will be scaled up, but basically it's an overnight E. coli fermentation. In our lab scale, we then harvested the cells, and I mentioned the DSP strategy earlier, that we want to have a relatively simple DSP, but also harness the heat stability properties of these proteins if possible. So how the DSP process for spycatcher aldolase looks like at this moment, and there are two options to do it, but we always need some kind of cell lysis, of course. We use the sonication technique in our lab scale, but it will or may go to high pressure homogenization eventually, I believe. The centrifugation step for separating the different phases. Then a heat treatment step at 60 degrees Celsius. We could go higher, but 60 degrees Celsius was sufficient. It is also, if you look at the capabilities that we will have in the larger scale, doable, feasible. At 90 degrees and so on, it will get much more difficult. Followed by this heat treatment step is another centrifugation to get rid of all those aggregates or the waste, let's say, and then we can actually already split it up. There is listed a size exclusion step here as well, plus an endotoxin polishing step that we need. This is the whole full picture, and we know this gets us very pure material, approximately 95% purity, with acceptable concentrations and endotoxin levels, for example. However, a lot of discussions also about that. We, of course, want to have it even easier if possible. So there were different batches produced. Some of these batches went through the whole DSP, and some basically stopped after the second centrifugation, after the heat treatment. And both materials have been evaluated in animal trials. And I think that approach will continue. Next slide, please. For the E2 SpyTech particle, the basic upstream process is very similar, a little bit different temperatures and different growth profiles, but eventually it's very, very similar, as you can expect. On the DSP side, however, there's a little difference. In those particles, we were not able to basically purify them by heat treatment. That was not feasible for us. So we went with the process that is described here. Again, of course, harvest and cell lysis. And then this is followed by an ion exchange chromatography immediately. You don't see the endotoxin removal step here because this is still under investigation, but so far results indicate that we don't need it because basically we are at low enough levels due to the first ion exchange chromatography. Anyways, so those processes were repeated 3 times at the 5-liter medium scale and upstream and downstream, and this will now be taken further into the large scale at a different site, not in Hanover anymore. Next slide, please. I want to also tell you what we did for the baculovirus platform for the viral antigens really briefly. We mentioned the Schmallenberg SpyTech subunit construct as well as the Rift Valley. The focus, as I mentioned earlier, was initially then on the Schmallenberg. We repeated or we scaled up also to the 5-liter medium scale, repeated it 3 times, saw a very comparable process. We saw very comparable process profiles and performance as we expected. However, as already mentioned before, and that was true even after further optimization, the medium scale yield was still very low. You see here initial results in the 5 liters less than 10 micrograms per milliliters. That's in the range of baculovirus, but it's low and it's also about 10 times lower than what we would have liked and expected actually. Next slide, please. Nevertheless, this is the material we had, and for the project and in the timelines we use this. Next step, please. Since concentration was low, but we can deal with it, we applied ultrafiltration hollow fiber concentration processes to this and also confirmed that this is feasible and usable. Next slide, please, or next bullet point. Since this is a baculovirus process, we also have to inactivate the baculovirus that is still in the broth. That is usually done by binary ethylenimine, BEI. That also worked and was confirmed. Next slide. And eventually that meant that we had produced about 12 to 15 liters of this antigen for further use, for further processing, and we stored it at -80 Celsius. That came up earlier before. -80, or why -80? Because that's the temperature we are sure it's stable. Everything else we would like to go to cooler, but we would need to do stability studies to really be sure that this is feasible. Same story for the Rift Valley. I mentioned before we had seed passage questions or decrease in antigen production with seed passages. That doesn't mean or didn't mean that we could not produce anything. We couldn't use high passage seed, but we could still use low passage seed. So we also did that 3 times at the medium scale for 5 liters, and basically the results are similar. Acceptable process performance, but low yields, meaning we would need to concentrate the material to be able to couple it to the MPSPs in a feasible way later on. Next slide, please. And with this, I will hand over to Ronan.
Thank you, Alex. So I think we've seen this slide, so let's go to the next one. Yeah, at the beginning of the project, we were still using an old generation cell line. So in order to improve the productivity, we identified and knocked out proteases that affected the stability of those antigens. So here you can see the different expression levels that were derived from the 8 protease deletion strain and the 9 protease deletion strain. So you see that here in the 9X protease deletion strain, we definitely increased the productivity over the 8X protease deletion strain. Altogether, we ran it into a 1 liter fermenters, we could reach a yield of 1,800 milligram per liter. And this was in a 5 days fermentation, which I believe seems to be very high. Just to compare it to other production hosts like the baculovirus, it means that it's like 300-fold higher. And I think for us, so it was in 4 days. So altogether, I think we were very happy because we managed to generate a good cell line that could produce those antigens. And we all know that those antigens to produce are very difficult because they are, in a way, the kind of artificial proteins. And we are happy that C1 can really tolerate all kind of type of proteins and can secrete it to the media and with the right folding, and by developing those strains also with very high stability. At the moment, what we are doing is that we are transferring the technology to Alex and his team in Beringer in order to develop it for high scale. It is important to know that in principle, our experience with C1 is that it can easily be scaled up. The highest scale that we produced in the past enzymes were 500,000 liter. So we believe for the doses that will be needed for the Schmallenberg antigens, we believe that we can reach easily the right production scale for the commercial production. Let's go to the next slide, please. Here, we also, again, we can show you, I think, what the first thing that was done at Beringer. You can see here that they could reach appropriate kind of production platform, protocol. And you can see again that the production seems to be very pure. And as we know also, it was very stable. The idea here that something which maybe we haven't mentioned is that the Schmallenberg virus antigen, the GCN, also contained the spike tag and the C tag, and the C tag is actually helping us to purify it in a very short time and few steps. And here actually the figure is really showing how the purification went on. If we go to the next slide. So again, here we were trying to express the Rift Valley fever virus antigen, the GN. And here we also use the same cell line, as you can see here on the Western blot gel. You can see very nicely that the antigen was produced at a high level. Here you can see that already by screening of only, let's say, 6 candidates or transformants, we were able to identify the right cell line. And it is important to note that we don't have to go like in CHO, for instance, not like in E. coli or baculovirus, to a transient. We, from the initial, we are integrated the gene into the genome, and we are having already a stable cell line. In this case, in 1 liter fermentation culture, we could show that we could reach a level of 1.24 gram per liter. Again, this kind of production level obviously can be then scaled up to a commercial level. If you go to the next slide, please. So I think the participants in this project really help us also to have additional collaboration, and in order to participate in, I think, the world combat against the current COVID-19 pandemic. And the tools that we have actually established help us to, in a very short time, in 3 months, construct an expression cell line that can express the receptor binding domain of the SARS-CoV-2 spike protein. And as I said already, to generate a stable cell line and a very stable product. The expression was done in 5 days from fed batch fermentation process, and actually with synthetic media. So altogether, it was a very low cost production media. It is also important to know that now we are also increasing the productivity by optimizing the process, reaching about 3 grams per liter. And if you think that the current demand of the vaccination against the SARS-CoV-2 is huge, and I think the only way to really be able to produce enough doses to protect people from all kinds of countries around the globe, I think this high productivity is a very important aspect of what we can reach. But it's not just the productivity, it's also about the quality. So from the beginning, all the tests that we did, all the ELISA tests, we could show that it's nicely bind to the ACE2 receptor. And already we did several mice studies that really showing very high induction of neutralizing antibodies. In addition to that, just now we have finished one study with some transgenic mice that expressing the ACE2. And again, we could show that that C1 RBD could generate full protection. Here on the bottom, you can see a very nice product that being produced in several days of fermentation. But on the right side, what you can see is that the purification also were done in a very efficient way. In this case, we could reach at about 97% purity in a process that confer 70% recovery. So I think this slide actually really show not what we could really learn from this very important project, the ZAPI project, and how we can really use this knowledge to something that is now affecting all of us because of the pandemic. So we can go to the next slide.
Thank you, Ronen. So it's back to Alex.
Yes. One slide about basically the conclusion of the evaluation of the upstream and downstream production platforms. Of course, we have the 2 components as described that need to be coupled or can be coupled via the bacterial superglue. We have different processes for the MPSP but also for the subunit manufacture for the subunits. They have, as far as we are concerned, a minimal number of steps. We are still evaluating if we can even reduce those further in the example of the spike catcher aldolase. However, if we look at the subunit production that was just also described by Ronen, it is pretty clear that the C1 expression or using the C1 platform is the key advantage here for, as it's described, for yields, but also DSP, purification. we don't have to compare those numbers again, but as Ronan described, there's grams per liter base yields, and in terms of surge capacity, that's what we need. And therefore, for further development, C1 now is or has been chosen as the production house for those subunits. Fast development, and I repeat again, sufficient amounts. And what's also very important then from an industry perspective is the reduced fermentation volumes. We don't need 4, 5, 6,000 liters. We maybe need 10, 50, 100. And we can use smaller systems and more flexible systems, obviously. And I think as it's written here, that's exactly what we want to have, and it's exactly in line with the ZAPI objectives. Next slide, please. However, the story doesn't end with production of those two different components. We also have to couple them, and we had to look at how do we do this? How do we do this in a little bit larger scale than, how can we scale this up eventually? Next slide, please. My colleagues at the Hanover site and also in Lyon and other sites, of course, have done this before, but we specifically looked then at those parameters or those goals that are listed here. We wanted to have, of course, a fast coupling, whatever fast means. We have to adhere to the data. We want to have an easy and single-step process if possible. And of course, we want to have an efficient process. We don't want to waste material. And it depends a little bit on the different MPSPs or antigens, but we also looked at different coupling ratios. How does the system behave if you put together different amounts of each of those materials? Next slide, please. And you can click once more, I believe. One more time, exactly. Thank you. This is only an example of different ideas, different experiments that were done. In this case, it was the Schmallenberg SpyTech produced by the C1 dyadic system, together with our in-house SpyCatcher aldolase that was coupled. To give you an idea what parameters we looked at, coupling conditions, it's mentioned below what kind of temperature, what kind of time, room temperature, cooler, 1 hour, 24 hours, somewhere in that range. We looked at the buffers, we looked at the different ratios of the component, and we looked at the mixing conditions for the coupling. So basically, once we move this from milliliter scale to, let's say, hundreds, thousands, or liter scales and higher, the hydrodynamics become a little bit more important, and we wanted to see, do we need to mix? How fast do we need to mix? And going into that direction as well. Next slide, please. The results that we obtained in our studies with the medium scale material from either provided by dyadic or produced in-house told us that in our hands and for the future production of vaccines, we need an overnight coupling to reach full coupling, the best results. And we will do this at room temperature. We had different materials, as you can imagine. We had baculovirus expressed materials, we had C1 materials. We looked at, initially at least, and I don't want to leave this out, we looked at the baculovirus materials as well. Inactivated material coupled, concentrated material coupled as well. But once we combined all those steps, the efficacy went down also. So that's another advantage for the, or disadvantage for this system, and advantage for the C1 that doesn't need the BEI inactivation. I think Jean-Christophe will talk more about the analytics later on, but to monitor this coupling process and to get some kind of kinetics, different analytics were used. For example, the JES analytics, analytic instrument, but also some others where we could not only basically see the beginning and the end, but we also try to get information about the process in between over the different hours. This was done for Schmallenberg. This was also done for the spy catcher Rift Valley, together with the E2 spy tech component. And basically the kinetics look similar in our hands. This is still not thousands of liter scale, obviously, with these materials, but we have identified conditions that are robust and that we can use to go can be used in the future. Next slide, please.
Thank you. So I will take over and finish this presentation on the analytical tools, especially on the specific aspects. So it will not be as elaborated as we have seen this morning for monoclonal antibodies, but focusing on the new aspects of a particulate or particle display antigen, which is quite new for the requirements. And this work actually has been done mainly by my colleagues at Hanover and in Lyon with Cécile Sigouayot, but we cannot present today. So the objectives of a QC assay for a vaccine is to confirm the identity, the amount, and the quality of the different components, and in our case, of a final form, which is the MPSP subunit complex. And for each of these three type of components, as you can see here, we need to confirm the identity, the quantity, and the quality. And the quality is actually the particle or particulate aspect. For the subunit, it will be identity and quantity, and the quality can be confirmed or monitored by using a VNT monoclonal antibody, so recognizing the right epitope. But the issue is a little bit more complex when you address the MPSP complex, because you have two different types of objects to qualify at the same time. The identity of a complex means that you need to be able to monitor that the subunit which is coupled to the MPSP is the expected one, and that you have also used the right MPSP. Even if an emergency case, there is no risk of mistake because you will work with only the two components. You need also to assess the amount of the subunit on the particle after coupling. And in terms of quality, it's always better that you don't have any free subunit in the final product, because then you have an issue on defining the right ratio between free subunit and the coupled subunit. And this is why it is better to use a specific coupling ratio, as we will see. But as for most vaccines, we'll use, of course, the standard QC techniques that are listed here for concentration, protein concentration, endotoxin concentration, identity, the antigen, and so on. Potency is done with the different antibodies and monitoring of amount. And eventually you can also use, because we have a physical or biophysical object, we can use electronic microscopy because it's more and more accessible, especially with cryo electron microscopy. So I will not detail that, but just focusing on a system that we have developed through a lot of discussions in the group and the project team on how to describe the MPSP complex with only two types of assays, which are biophysical in nature and orthogonal in order to define or describe the full complex. So the first one is, and the two techniques are actually relatively new. The first one is A4F for asymmetric flow fractionation field fraction that will provide information, and we'll see details in the next slide, on the particle population homogeneity. It gives a particle average molecular weight if you have an homogeneous population. And it can give also, through just computing, an indirect information on the coupling ratio, but no precise information on the quantity. Whereas if we are using mass spectrometry and quantitative mass spectrometry, and you will see details also in the next slide, you can access the full identity and amount of each component, the MPSP monomer, the identity and the quantity of the subunit, which is coupled to the scaffold. And then you can deduce a direct or directly or indirectly, in fact, the coupling ratio. But with this technique, you will not have the information on the particulate feature of the material you are analyzing. So, but combining the two, you can get a full picture of what you want to see. So the first new technique is this asymmetrical flow field fractionation, which is a kind of laminar flow. It's a liquid HPLC, which separates particles based on their size and their weight. And you have a cross flow in a capillary that will move the particles out of the main flow. And then you have sensors on each side to detect the particle and gives information on the size, the diameter, and the molecular weight, because it's all calibrated on the molecular weight. And what is remarkable is that this technique is extremely precise, using materials or MPSP complexes generated at FLI, but by Andrea Ebischer, a PhD postdoc in the lab of Martin Beers. We compared what was designated as unsaturated particle versus saturated particles because different ratios were used for the coupling. So one subunit for 1.5 MPSP monomer, lymazine synthase. And for saturated, there was a big excess of subunits, 3 subunits for each monomer. And you see that for the first population, we could see that we could monitor a molecular weight of 2,651 kilodalton, corresponding more or less exactly to one scaffold, lymazine synthase, coupled to spike catcher, and 30 subunits of Schmallenberg GC head here, based on the molecular weight that we know, of course, and the size diameter average of 20.4 nanometer, whereas with the saturated particle population, the molecular weight measured was bigger, as expected, 3,370 kilodalton, corresponding to 50 subunits in this case, coupled to lymazine synthase. In fact, lymazine synthase is the smallest of the scaffold we have used, and due to steric hindrance, we think we cannot reach the full saturation, that would be 60 subunits coupled on the scaffold. And we will be repeating that very soon with materials produced at medium scale and large scale. Second information is the quantitative mass spectrometry, which has been developed recently. It is based on the specific digestion of your targets. So you provide, you generate peptides, you verify, so you select one peptide which is providing the unique identity of the target, and you synthesize a corresponding peptide with an isotope that will be spiked into the material to be analyzed. You know exactly what you spike with in terms of amount, and you just compute a ratio between the labeled peptide and your target peptide. And from that, you can deduce directly the identity of each component, and you can do multiplexing. So you can measure in the same channel, you separate different peptides coming from the subunits and the scaffold. And at the end, you have a full information on both components, and also providing you the information on the exact ratio that has been performed. A conclusion on this analytical part is, of course, we use also specific reagents, and the team in Hanover has been really remarkable in being able to generate anti-spike tag and anti-spike catcher superglue antibodies. I was thinking it was not possible, but they did it. They can be used to monitor as a generic tools, which is kind of heresy, I would say, in the QC world, because you use a non-specific tool for measuring something which is specific, but due to the fact that you are fusing the target with the spy tag or superglue moiety, you can still monitor your target. We think we have developed a very innovative analytical approach driven by the biophysical nature of MPSP, and the first time we can really have a stable, very well-defined object to measure, while most of the vaccines are some kind of black box you don't really understand, at least for inactivated vaccines, not for live, of course. And eventually we can go to QC techniques which will not require specific antibodies. So I don't know how this will be acceptable by regulatory authorities. There is also a change of mindset in this domain to be generated. But for the first time, again, we have 3D structure to monitor. We have also a quality, a built-in quality design, because the coupling is not inducing any constraint between the subunit and the scaffold due to the very easy and simple coupling of the bacterial superglue. And ideally, maybe optimally, eventually, all key information can be obtained with 2 orthogonal assays only. But maybe that's a step which is too big to jump right now, but I think the potential is clearly here. And also to finish, I want to thank all the people in Lyon, in Hanover, and at Dyadic who contributed to this part of the work in a very difficult year for many of us. Thank you. So we can go now to the 2D session. So I don't know who will drive the slides.
Shall I share the slides for you?
Yeah, that would be good.
Yeah.
Okay.
And that will be a shared session between Jeroen, no, sorry, Paul, Martin, and me.
Yes. Shall I start?
Yes.
Yes, this is Paul Wiegerseur from WBVR. And in this part, I will focus on the validation of the MPSP particles used in a LAMP vaccination trial. So next slide, please. And the objective of this work was to validate the GN head subunit produced in using the baculovirus system coupled to lumicin synthase, and to really assess the influence of coupling. And in the LAMP trial, we vaccinated twice with 2 weeks at a 2-week interval, and challenged the animals 2 weeks later. And this was in a collaboration with the FLI in the group of Martin Koschub and Markus Keller. And following a challenge, animals were monitored closely. And as you can see on the right, there we performed a GN head ELISA and a virus neutralization test. And starting about 1 week post-vaccination, antibodies are induced, and you see some increase of some higher levels of antibodies in the coupled group. However, at the time of challenge, antibody levels seem to be similar. Also, the virus neutralization test antibodies were similar. However, next slide. If we then look at the body temperatures as a readout of the challenge, you can clearly see that mock vaccinated animals induce high fever, and there is some temperature increase in the uncoupled group, and this is also reflected by the viral RNA in the plasma. So in the group which received the lumicin synthase plus DN het, so uncoupled, then there were a few animals which still have viral RNA in the blood, and also presence of virus in the liver and spleen samples. In contrast to the animals who were vaccinated with a coupled version. So this looked very well. So there was improved protection in the coupled group. So next slide, please. So following this experiment, we at WBVR performed a similar trial in which we assessed whether different MPSPs might also influence the immune response to GN het. And we used lumicin synthase, albalase, and E2 and coupled the same GN het subunit to these particles and used this in a vaccination trial again. So next slide, please. But this also involved lambs and with a similar regimen as well. And the ELISA and VNT results clearly show again that there is potent induction of a very efficient induction of antibodies also after the first vaccination, and that at the moment of challenge, that there is no significant difference between the different MPSPs. And this is also reflected by the viremia. So this is on the next slide, in which, and body temperatures, in which we observe that again, mock vaccinated animals develop disease. So they present with increase in body temperature, have very high levels of viral RNA in the blood in these animals, and also a lot of viral RNA of infectious virus in the liver and spleen samples. And in none of the vaccinated animals, any viral RNA vaccine of a challenge virus RNA was detected, and as well as not was detected in the organs. So this looked very well, but was not yet conclusive whether one MPSP might be better than the other. So next slide. To conclude on this, we can say that all MPSPs or all complexes were well tolerated and safe. We did not observe any side effects following vaccination. We showed in the first experiment that the coupling improves vaccine immunogenicity and efficacy, and this has also Jeroen in a few hours ago presented data about the mice experiment, but this is with lambs, and these are natural target species. So we now also show this for this important species, and we show rapid onset of neutralizing antibodies, also detectable after one vaccination already. Highly efficacious, so sterile immunity was observed, and I think the MPSP selection can now also be made mainly on the manufacturing process. So as we observe no major differences. So with this, I would like to hand over to Martin Beer. He's going to present a part on Smallenberg vaccination.
Oh, thank you very much. So I will present the data which are now made with the material lumicin synthase. On one hand, that was our first trial with the scaffolds, and the SBV GC head or GC head stalk. And the GC head stalk, you see it on the right side, that was produced in the dyadic C1 system. And the GC head was produced in the S2 cell line by Andrea Erbischer, and she also coupled all this with the lumicin synthase. So if you look at the left side again, you see that we have their electron microscopy pictures of the LS lumicin synthase with the head. Then we have the C1 expressed head stalk with the LS. And we also tested in this trial what I mentioned before, the directly fused peptide, because we wanted to see if there is any immunogenicity in the peptide which was reactive. This was given to 4 animals each. That was calves. And the challenge was done with infectious serum, because that's the best challenge for Schmallenberg. And nearly all challenges we did with sheep and with cattle are done with the same infectious material. And there we had the possibility to compare this to a naive control group. And on this, we used also mainly the unsaturated particles, which is a big advantage, as you heard before, for the production. Next slide, please. So if we look at the neutralizing antibodies, then you see we had the scheme of a very short booster in immunization after 14 days. So until 14 days, due to the first vaccination, we did not see neutralizing antibodies. But if you look at the right graph, you see that there were specific antibodies seen in the ELISA. The ELISA is a little bit more sensitive, and I'm pretty sure that if we would have waited a week longer or so, we would have seen also neutralizing antibodies. After the booster immunization, we see very nicely coming up both the neutralizing antibodies and the ELISA Qc specific antibodies. And as we saw before, the head-stalk construct is better, inducing higher neutralization reactivity than the head alone, but both are good. And we don't see neutralizing antibodies, measurable neutralizing antibodies, if we use the peptide alone. So even presented on the lumacine particle, it's obviously not enough in this case. If we look then in the challenge experiments, next slide. Then you can, on the left side, see that's a black curve. This is the classical viremic curve of the naive animals. You see a reduced viremia, so there is some protection, but not a lot in the peptide immunized animals. And there is full protection in all the animals which we had immunized with the Qc head and the Qc head stalk. So no viremia, and I think even more important, no SBV RNA detected in tissue samples. So normally you find later on, for example, in the tonsils, in the lymph nodes, you find RNA. This is not in the ones getting the head or the headstalk construct. So a very high level of protection, which we can call a sterile immunity. Next slide, please. So this was the first proof of concept in with the particles, and also using, for example, C1 expressed protein. In a second step, another scaffold is used, and it's again material produced with the SPI-tag, and it's the headstalk domain, because this is the one which is superior, and this is expressed again in the C1. And the objective this time was a dose range evaluation. So use 50 micrograms down to 5 micrograms, 1 shot versus 2 shots, and also some safety issues concerning this size exclusion cleaning up. So this means no SEC, no further cleaning, secondary cleaning, or with the additional purification step. And this was done in lambs at Boehringer in France. Next slide, please. And here you can see immediately that there is, if we look at the Qc ELISA antibody kinetics, a very nice reactivity. This time we used a longer term between the two vaccination applications. And the second one was given at day 28. but after 14 days, again, the animals here reacted very nicely and more or less independent of the amount of antigen. So there's no big difference after the first shot, and it's clear there is again a booster if you have the second shot, but also here the booster effect is more or less not markedly different. So that I think there is no significant difference between the different dosages. So 5, 10, 20, 40, 50 microgram reacted more or less the same, and there was no difference if there was a purification step or not. So I think very impressive results that this presentation of this highly immunogenic key antigenes is very, very efficient, even with very low amounts of antigen. Next slide. And this is at the same level seen with the virus neutralization titers. There we see that even after one shot, some show reactivity with 40, 80, 1 to 40, 1 to 80 titers, and a very nice booster in the neutralization with some differences. But if you look at the 2 times 5 microgram, the blue line on the right side, this had a high titer, and this is even not titrated to the end. So some of these values might be above the 1 to 600 titers. So we are working on this now. So this is very new results, but I think it shows clearly this works, and even one shot shows some efficacy over the time with 20 or 50 microgram. Next slide. So the conclusions for this dose range study are that all immunized animals had detectable neutralization antibodies. So this combination antigen, particles, and adjuvants worked very nicely. There was no impact of this non-purified versus purified scaffolds. We more or less saw no dose effect between the 50 microgram and 5 microgram, which is very good news for the production side. The neutralizing antibody responses are similar after one shot with either the 20 or 50. So again, here the amount is probably not the most important. And the titers are still there, also neutralizing titers at day 49 after a single shot. So that's also something we had not seen before. And I think all these titers are in a level where we know from previous experiments that they should be very well protected against challenge infection with Schmallenberg virus. So very, very promising. And therefore, we decided to start an experiment in cattle. Next slide. And that's ongoing. So last Monday, we had the booster vaccination. We have 3 groups with 5 calves. So Kerstin Wernicke is the one and her team doing the study at the moment. So there was immunization 1 and then immunization 2 for one group. So we have a single immunization group, a double immunization group, and we have a SPV subunit dose, and it's 10 microgram, 40 microgram for the double 10, for the single 40, and the PBS control group. The challenge will be performed at day 35. So I think we are believing that it will work, but you will see in a few days. We know we will do PCR again in the blood, and at the end of the trial, we will look at tissue samples, especially the lymph nodes, to prove if there is complete protection or not. Next slide. So some conclusions before I hand over, but I think they are all these vaccines look really, really good and safe in the target species, highly potent and efficacious. We see sterile immunity Rift Valley trials see it. And I have not shown this in Erwarte-Schmallenberg, but you see a very nice differentiation because it's only Qc specific or the head domain specific antibodies, and there is commercial assays for the nucleoprotein. And you can immediately differentiate between infected or vaccinated and infected animals. So there is a DIVA strategy immediately implemented. And with that, I hand over to Jean-Christophe.
Yeah, next slide. In fact, also to thank all the people who worked on these animal experiments, especially my colleagues in Lyon, Mathieu Chevalier and Brittany Guillaume, Alexis Parizeau for the adjuvant, and Cécile Siguayo for making the coupling. In fact, as an example of the robustness of a coupling process, this has been done by different people in different labs without training, and it works as long as you follow the conditions and the requirements. I think, as you have seen from these experiments in target species, that it looks like we are entering a new world with a particle display antigen. We need to still explore how low we can go with the vaccine dose, at least in the Schmallenberg model. Of course, it will not be possibly the same with Rift, but it's still very impressive. And actually, when you look at the papers currently published with the ZAPI-like approach for SARS-CoV-2 RBD, and the papers mentioned by Bart this morning, you can get very good responses in mice, of course, but with 0.1 microgram. So there is still room to improve, and you can understand also from a manufacturing perspective, when you see on one side the yields we get in C1 and on the other side the low yield we can get on the when we present the antigen of the scaffold, but we can access a huge capacity in terms of vaccine antigen. The issue to solve now is to produce the scaffold at a sufficient level.
So perhaps before we start, I think it's good to remember that it's about 5 years ago when we really were involved in writing this project. We could never have imagined the successes that have been gained in all the fields that have been tackled. So what I'm going to do is a daunting task, I think, really going to summarize what has been achieved with the whole vaccine efforts that have been done in this particular project. And when you look at this slide here, what you quite clearly see is more or less a summing up. I will not go through all the details, obviously, but just to give you an overview. We, first of all, selection of key immunogens for each of the target viruses. So that was done by antibody or monoclonal antibody analysis, peptide analysis, crystallography, especially for the SBV and Rift, and identification of the most immunogenic subdomains, production of the key immunogens in suitable expression systems, as we have heard, in vitro characterization, and then the studies in animals for basically for immunogenicity and for efficacy. And that was done for the 3 viruses that we had selected initially, and we could not have guessed that especially the coronavirus would become so important in all these studies. And all the studies confirmed the immunogenicity and efficacy of the selected antigens, and all the selected antigens represent major immunogens of the respective target viruses. So basically, the final candidates, as we have heard, it's interesting that when you talk about the Bunya viruses, you have on the one hand the Schmallenberg and the other hand the Rift, and it's 2 different domains, as we have heard, that were identified. For the MERS coronavirus, as we have heard, it's the S1 domain and the receptor binding domain. So when we move on, then when we hone in on the proportion of the immunogens, the essential parts, we have seen that for the Schmallenberg is a small part of the GC head stalk, and for the Rift it's the subdomain 7, and that's about 314 amino acids of it. For the MERS coronavirus, it was the receptor binding domain itself, yeah, and it was shown that it's much more immunogenic than the whole S protein or the S1. But the interesting thing is that, of course, if you go for the receptor binding domain, as we see at the moment, all these mutations that are popping up, there may be a problem, and we may, both for the usage of antibodies, monoclonal antibodies, but also for vaccines, that we may have to go for a combination of the two. And so it's interesting also that this information can be of key importance also for the mRNA vaccines that are being used to date in the selection, especially of the subdomains that are necessary or minimal parts of what we have to use. And I think this is really an interesting area to go into. And I think that it has been shown also that the rationale is based on industrial manufacturing aspects, and the yields are much higher if one uses a relatively small soluble subdomain compared to the whole thing. But we also discussed the possible drawbacks. What we did not discuss is a possible enhancement. I won't talk too much about it, but we know from the animal models, but also for the coronaviruses, that enhanced disease induction, both by antibodies and by vaccine, is a thing we have to be careful with, and especially not with the Bunya virus, but definitely with the coronaviruses. So when we move on then, and we look at the we heard about the baculovirus system. It was quite promising, and it was quite successful in the whole thing. Yeah, it worked pretty well. And I should say immediately, when we look at the industrial world, there's a lot of experience with baculovirus-produced vaccines for pears, for SERCO-2 in pigs, but also the flu block, and the first recombinant vaccines for influenza in humans, which is doing very well. So there's a lot of experience with that particular presentation. And we need to know why, as was presented quite clearly today, it is not always as efficient as we would like it to be. And I think there is an important point there that we cannot, we do say with the expression systems, we have kind of a model system, but it should also be realized that you can never put all your cards on one system, because for reasons we don't always understand, we might run into trouble. Yeah, and often it is, as you see here, is a trial and error approach. So basically, when we look into the baculovirus system, and you've heard that explained extensively, yeah, we need to understand the complexity of the protein that should be made, the properties of the vaccine, and the highest productivity in the baculo system, as we have heard today, it was not, for the viruses that were looked into, was not optimal, and it went up to 20 to 90 milligrams per liter for the Bunya viruses. Well, if we go on then, and then of course a lot of attention was being spent to the dietic C1 system. Before we go on, I think we should realize that it has not been used in humans for vaccines yet, but that we'll come back to that later when we talk about regulatory issues. And also is important the collaboration with dietic on the one hand, but also with the Boehringer group actually eventually resulted in an improved C1 strain and process that has been used now for the SARS-CoV-2 RBDs, the receptor binding domain subunit. And if you look at the figures, and Roland has explained that quite well, you go up to 3 grams per liter, as he showed, with 98% purity and 70% recovery. So these are impressive figures if you compare it to the baculo system, for instance. There's other opportunities, obviously. And then C1 is being investigated for the next generation products of SARS-CoV-2 vaccines. I'll come back to that. Well, the project as such is very flexible. It takes only 2 months to express a new RBD variant at a significant level. This is, of course, at the moment when we are talking about SARS-CoV-2, yeah, and all the experience came from MERS initially, of course. But when we look at SARS-CoV-2 and we see the mutations popping up, the UK mutation, South African one, South American one, then we realize that basically it would be very helpful if we would be able to replace RBD variant very rapidly. And then the question again, should it be the RBD alone, or should it also be the, or should it be the S1, or anyway, the spike glycoprotein? Well, some very interesting data presented by Bart Haagmans also. You see here mouse studies with the C1 expressed, and now with the SARS-CoV-2 RBD vaccine candidate. And this is in a study where in mice, he looked to what extent neutralizing antibodies, ELISA antibodies, but also neutralizing antibodies were being produced. Quite promising data, I should say. And following up on this, we have started experiments now together with Bart and Beren in Hanover where we have used this particular antigen to immunize in different forms, to immunize hamsters. And we looked into the coupling with nanoparticles, the high dose alone, or adding of an adjuvant, so all the different parameters. And quite interesting data. That the data is not complete yet, but it actually indicates that we may be preparing for a human phase 1 trial after toxicology has been done with the selected project, of course. So going on, we were talking, of course, all the time about the superglue systems that were presented, self-assembling, temperature resistant, stable, and I'm not going into all the details again. These scaffold particles, and it's all about multimeric presentation. And with this very stable system that has different possibilities, as we have heard, also with the C1 fungus expression system. So there are many examples there of how you can put this thing together. We know, of course, that if you go for those very small peptide-like antigens, the immunogens, that the immunogenicity of those structures themselves are very, very limited. So you need the multimeric presentation, and this seems to be a very robust system, as we have heard. So some examples here with the E2, with the E2 SPI-TEC system here, with the aldolase and also with the LS. And you see here some of the data here with Rift and Schmallenberg. Yeah, so the antigens that were produced in Bachelor on the insect cells here, they may contain the strep tags in addition, and antigens were also produced in C1 cells, both by Dyadic and by Boehringer, as I said. Well, these multimeric presentation forms are currently aldolase and E2. That has been selected now, produced in E. coli, and we have heard the data there, which were all quite promising. And I think the interesting thing here is what we have seen is that you really go from the drawing board all the way to something that's being tested in animals in a very flexible platform. So basically, if you look here, the MPSP expressed in E. coli, and the coupling there, they worked on the ratios. You see the different types here, ELS, and now the E2 and the aldolase being more favored. And then you see the coupling details here. Mixing the two components. The interesting thing is that you can basically the scaffold, you can already have that one ready in large amounts. And then it's more or less like adding an adjuvant with a very simple system. And the system also works whatever orientation is used, and there is no interference of the scaffolds themselves. Well. Boehringer used different systems, different processes, and developed new analytical techniques, as we have heard from Jean-Christophe, which he quite nicely explained. And a lot of animal studies have been done in the meantime, as we have seen. Good. So when we look then at some of these, when we look at search capacity and search in relation to the platform activities. So it's expression of the immunogens in either Bachelor or C1 system, and the MPSPs in E. coli. There are different possibilities also to swap the things. And here you see coupling ratios for generating these complexes, analytic analysis, and animal studies in the target systems have been done. And continuing along this line, we see here that let's say the project did not encounter any technical failures or major delays which were harmful to the project as it is. There's no project that will go from 0 to 100, without any hiccups. And the only delays that were encountered were due to the COVID-19 outbreak, the pandemic, and also a reorganization in Bergen that happened during the project. And so that caused the project to be extended by 1.5 years. But I think it's if we look at the overall result, there was no harm done. And the critical goal was to show the robustness of the platform, and I think that was met. An added value of the project would be to communicate and how the decision process was done, where if you get hiccups, how to overcome them. Well, we could extrapolate this this process to other pathogens. Of course, relatively simple, I shouldn't say that, but at least pathogens that can be inhibited by antibodies alone, which is not true for all the pathogens that we may be facing in the future. And if it were to work for African swine fever, that would be fantastic, of course, but that's probably not the case. And it would be useful to do a comparative development in quality of the antigen and yields of different expression systems using the same antigen. And for a better added value, it's necessary to perform a benchmarking with other processes, obviously, and identify the benefits and the improvements of the process. But I think overall, we can say that it has proven to be, in the hands of different people, quite a robust system. Well, it would be interesting to target more complex pathogens. The choice to not target the complex virus was a chosen limitation of the project. Really to focus on certain zoonotic viruses when we wrote up the proposal. And this whole thing will be discussed in a ZAPI white paper that is going to be made. Well, then the other thing is, of course, that in times of epidemics and pandemics, it is important that we also have access to a fast-track regulatory process. And definitely when we are facing a situation where the antigen is changing, we get mutations that cause escape. And so there is a need for strong European support for vaccine development and production capacity. And especially Joris van the Putters Group, he has been doing a lot of work on really going after these regulatory things, a platform master file. So basically, it's interesting that in the veterinary world, the veterinary world is more advanced here, and basically that's a lesson to be learned, that a lot of these things can be extrapolated from there, and the immunological medical products for human use. There is a core document now shared with EMA. Well, there are intense exchanges with the EU Commission and the EU Parliament for the fast track procedure being covered by Article 25, applications in exceptional circumstances, and basically the discussions also with national regulatory agencies are ongoing, and especially requirements for monoclonal antibodies produced in transient systems, and also the 3R policy getting a lot of attention. So when we are talking about this platform master file, there's close collaboration and consultation with ZAPI, sideline effect with EMA and FDA. They're also looking at the example of the veterinary legislation and suggestion to discuss with the backbone platform, what the backbone platform can offer. And obviously we have the example of the human influenza vaccines, where a new vaccine is being made twice per year, on the basis of molecular and epidemiological data. So that's quite an important thing. And for some of these agents, and especially when we are talking about the coronavirus that's circulating, that might be an interesting thing. Well, dissemination, and I went the other day, I went through everything that has been published and presented, and to be honest, it was too much to really file that all. It is in all the paperwork. But if you look here between April and October, last year. ZAPI resulted in more than 35 presentations, more than 10 manuscripts. And you see here, there is representation at different meetings, the nidovirus meeting, the Bunya virus conference, the One Health conference. There was, so there is a lot has been done. And of course, we are talking now during the ZAPI stakeholders conference. ZAPI is connected to several master teaching courses to stimulate the new generation to go into this, and it's that is organized or participated by Boehringer, and here we see the H2020 Erasmus Mundus from Hanover, a collaboration between the veterinary faculty and the medical faculty, and then we have the ZAPI modular vaccine methodology is currently being applied to SARS-CoV-2. As I said, some of the antibodies of MERS-CoV were approved to cross-react with SARS-CoV-2, and broadly, as Bernd Jan said, broadly reactive antibodies against the coronavirus is one of the goals there, and these are gearing up also for clinical trials in the meantime. ZAPI has also led to the establishment of additional projects. There's a lot of different projects now. There are the ZAPI project. There's discussions with BARDA ongoing, yeah, and 2 SMEs are mentioned here, but already the involvement also of Artemis Viral Clinic. So there was a lot of spin-off there. Well, in summary, when we look at the platform technologies, there's development of an innovative platform for the rapid characterization of key immunogens and the development of neutralizing antibodies corresponding to these immunogens. Preclinical testing, decision tree-based system has been developed, and testing the small, medium, and large-scale production of these new findings. The manufacturing processes here, there's the efficient manufacturing processes are being developed. I specifically would like to mention Dietic, the C1, the fungal process there, which gives enormous amounts of antigens. And I think, especially in the face of the world situation with the pandemic, a system where you can produce much more vaccine would be an ideal one to look into. So if that is if that were to be the only spin-off, that would already be fantastic. Well, the development of full in vitro assays for quality control of the candidates, and setting up and proof of concept of the analytical tools for the production process monitoring. So it is really a very complete system altogether. And then, of course, I talked about national regulatory agencies and the EU authorities. There's a lot of discussion ongoing. We'll hear about that more during the meeting also tomorrow. And then especially the platform master file there, and how to see what can we learn from the new regulation on veterinary medicines, also for the human acceptance of new vaccines and antibodies. And so I think that is quite an interesting thing. So if I can make an overall summary of all the ZAPI vaccine achievements and challenges, is really the development from a concept, from the drawing board to an industrial process. And ZAPI has been a major success, I should say, in terms of the scientific data that have been generated, the industrial results, yeah, a system for better epi and pandemic preparedness, apart from the regular vaccines that need to be made. And then, of course, the PMF that I mentioned before. It has led to very tangible and practical outcomes. New projects, a whole series of new projects has been initiated, development and collaboration with a number of different small, medium enterprises. The partners have been enormously resilient, and I should say the partners have collaborated in a way that I've never seen before. This was really an example of how such a project should be run. Well, ZAPI, of course, has the obligation now to communicate and explain the successes of the consortium, and also of the decision processes that were exercised. The selection of viruses. So basically, I remember the early discussions on the project that we said we need to go into a non-competitive area, so where we can quietly work. Well, it turned out to be quite different. And for the future selection of viruses which can cause a pandemic or major epidemics, yeah, it's quite an important thing, and we have to define those to see how we can implement the system. And obviously, it's quite important to also for the future to create these kind of projects to really be better prepared. Yeah, perhaps a last minor point that no, not minor point, perhaps a last point that was less of a success. If you look at regulations on animal experiments, that needs to have more attention. If you look at the regulations in different countries, even in different länder, in for instance in Germany, then we see that that it can be quite inhibiting to for the development of intervention tools if there is a blockade or it's very difficult to get your animal experiments permitted. But again, that's very difficult per country. Yeah, but I think it is an important point to take into account as well. Well, stopping with this this last slide. So basically, I think it's fair to say that it was a fantastic project. I would like to thank you for your attention. And there is now room for a question and answers session, meaning that we basically we can go for questions and answers. Two ways of doing it. You can either go to the chat box and deposit your question there, and the other way is to raise your hand. There is in the system you can raise your hand, and then that's a way indeed to start the discussion. So I would like to leave it there and open the floor for discussion.
Thank you, Ab. So you can ask your question indeed through the chat box or directly if you raise your hand.
Perhaps Jean-Christophe, to start a discussion, I could be a little bit provocative there, and I would say the viruses that have been chosen from the beginning, yeah, so we consider them to be the easy ones, so to say, which that didn't turn out to be completely true, but in terms of what we could expect, yeah, if we are facing, for instance, if we were to use the same technology that we discussed today for a virus like African swine fever, I mentioned that, or for HIV, those kind of challenges, what would be your reaction there? Because I only talked about all the successes, obviously.
Yeah. Just to remind that, in fact, the ZAPI project and the call was very specific for addressing zoonotic diseases because it was IMI, so we could not go only for animal or veterinary diseases. So the choice was relatively limited, and the objective was really to achieve this surge capacity manufacturing in case of a pandemic or a panzootic. So reaching a huge scale in terms of number of doses. And you don't have many viruses doing that. So the acute viruses indeed are, for me, and you know, that are quite easy to control in terms of vaccination, immunization, and especially through antibodies. And I will comment that later. And the second parameter for the choice was that we didn't want to go for impossible targets like HIV or African swine fever. So in order to confirm that we could be at least as good or better than the existing vaccines. Because chasing two targets at the same time is quite difficult when you are building a new platform. So I think we indeed achieved a lot of success, and we opened new ways to think about vaccines. Now we can apply that to other targets, but that would be a new project, not ZAPI.
I agree, but let's say if you're talking about zoonotic pandemics, then HIV is a zoonotic pandemic as well.
Yeah,
and
but indeed, but you have time. You have time to react. It's not spreading so fast.
Yeah, I agree.
Compared to COVID-19.
I agree. Okay.
Maybe a comment on the antibody, because indeed that's the classical way to monitor the quality of vaccines, and it's a tool that is easily measurable and classical, well known by regulatory people. So it's everyone is comfortable. Of course, it would be good to have also an assessment on the cellular immunity response or innate immunity. But for me, based on my experience, past experience with VLPs, not the scaffold, it's never black and white. You have always some kind of cellular response. It's not only pure antibody or pure cellular. It's always a mix between the two, which is variable depending on how you present the antigen to the immune system and how which adjuvant you are using also. So it's a complex equation. But with this particle display, I think we can learn a lot because we can be very precise in the way we trigger the immune response. And in fact, the particulate aspect is important. If you remember what Paul has shown in terms of a comparison between the groups where the scaffold and the subunit were just mixed together but not coupled, you could see there was still a quite interesting effect by the presence of the particle in the vaccine. So inducing some kind of better response, better stimulation, and you could see, you could think that MPSP can be used just as an adjuvant for some vaccines, and that's maybe a way to explore in the future. More complex to manage from a regulatory aspect, but definitely there is an immune impact that is not neglectable.
I agree. I think we shouldn't make it more difficult than it is. But on the other, I see a lot of questions coming in, especially about pox viruses at the moment. Yeah, so would there be a case for using the system for pox viruses? Is the question.
I think maybe I can briefly comment on that.
Yeah.
Yeah, I think we should also, of course, if you find like a real key immunogen on African swine fever, for example, that would like be super efficient in neutralizing the virus, which I doubt exists actually, then you could use the same technology. But I think we should also be realistic and not claim that we would have a platform that's applicable to all viruses. That's just not realistic. I think it would be good enough, and that's also why we decided to have this focus within ZAPI, is that most zoonotic pathogens that we know are enveloped viruses. They have glycoproteins. Neutralizing antibodies are at least important for protection. So I think that was a just decision to make. But nevertheless, looking at other platforms for other pathogens is of course equally important. And I think maybe the innovations that are now being made for SARS, maybe they will surprise us also for these pathogens. We don't know. But I think personally for African swine fever virus, I would bet on a live attenuated vaccine, definitely. But that's my opinion.
Especially because the T-cell immunity will be quite important. There's a lot of questions about that. But here a question that comes back here also is about on the one hand, the mutations, the mutations within the receptor binding domain of SARS-CoV-2, but also the glycosylation, of the C1 system. So is the C1 system really the best system for everything? And I think we clearly said already that that's definitely not going to be the case. But then glycosylation, perhaps Bernd Jan, could you say something about that? Or Martin is.
Yeah, I think glycosylation for the vaccine antigens is something to look at. It's of course different compared to expression, the glycosylation in mammalian expression systems. Particularly if you want to use the expression system for antibodies, then glycosylation is key because this plays a role also in Fc-mediated effector functions. But for vaccines, I think it is similar to the baculovirus system, right? So in that sense, and the baculovirus system has also been used for human vaccines, as you indicated, for flu block. So there might be less of an issue.
that was always an issue of discussion, that it would never work because of the glycosylation. And then, as a matter of fact, upon looking into the results of the human vaccine data, it actually is no problem whatsoever. And even there are some indications that immunogenicity is at least as good. So so basically, there's another couple of questions that come up about the adjuvant. When you use this multiple presentation systems, so these nanoparticles, these scaffolds, does an adjuvant add to that? Do we need an adjuvant as well? So in principle, you can ask the question. I remember that was also for Flublock. That was a question. If you were just to go for let's say 3 times as much antigen instead of the multimeric presentation, instead of going for an adjuvant, if you now were to use just a large amount, because we know that an adjuvant gives an extra complication. The multimeric presentation as such is an extra step in the production. So what is the answer there? Perhaps that would be a question for Jean-Christophe.
Yeah, I can start. Indeed, that's still an open question for me. From 2 perspectives, you have to understand if the adjuvant is adding something on the stimulation of the immune response. It looks, it is, but it's also a perspective of a payload and eventually of a manufacturing capacity. So if we remove the adjuvant, you can probably still have a very good response if you increase the payload, but it means that you will reduce your manufacturing capacity. So that can be done for some targets, but not in a pandemic context for me, or at least panzootic for animals. There is no issue to use very safe and registered commercial adjuvants. For humans, yeah, for people, it's still an open question which adjuvant you can use. It would be interesting to see for SARS-CoV-2 if just aluminum gel can work, or the F59 from GSK.
Yeah, we happen to be in the middle of a hamster experiment where we put all these things in a row. Yeah? So looking at more antigen, and for basically it could well be that with the C1 system, more antigen would be a practical thing to do.
The same, indeed. When we see today the yields that can be generated by the C1 system, you also think differently from before, because there was a limitation in the supply of antigen, and this system is removing this bottleneck, so you can go for higher payload with no effort.
Exactly the same question arose when we were discussing about Flublock, and then there was really discussion about an adjuvant or more antigen. And since the Bachelor system was so efficient, was considered for that particular hemagglutinin, it worked pretty well. And then it was decided not to go for an adjuvant. So I think we keep all the options open there.
Yeah, it's also linked to the nature of the immunogen you are using. You know that some are very immunogenic and some less, and you have to play for the same amount of protein, that's still a big unknown in immunology and vaccinology. The day we understand that, how to increase the immunogenicity of a given target, I think we'll solve a lot of issues.
I see a raised hand of Joris already for some time.
I see a hand of Martin first, and then
Martin.
Joris came later. Martin goes first then.
Well, no problem. One comment to the adjuvant. So we compared that for the Schmallenberg virus in the mouse model, and there's no reaction without the adjuvant. So I think at the moment it's difficult. For the flu situation, I think there you have an additional factor that you have this natural immunity. So there is even a vaccination can be a booster, and therefore it can work without. But if you look at the pandemic vaccines, you normally also use an adjuvant because it's an initial primary immunity, and then I think it's difficult. I also had hoped, and I still think we should test it for other antigens, but without adjuvants will be difficult. And to the other questions, I think for me, what we mainly learned is that we can have a rational view on the selection of the domains. So also for African swine fever or for LSDV, lumpy skin disease virus, which is also a pox virus, we look now with a different way on the different proteins, because to express some of these proteins in the large DNA viruses is not really possible, but domains can be expressed. So we start there also, and others do the same way, having crystal structures or other analysis to find the domains and even combine domains, for example. So you can think of 10 immunogenic domains of these large viruses, which give more immunity than the whole virus at the end. So for me, mainly in the last 5 years, the complete view on how to select antigens has changed. It's not taking the whole protein, and that's always the best. So I would not say this anymore. So it could be, but there could be a domain or an immunogenic part which is even better, easier to produce, and provides better immunity. So I think that's, for me, important to mention again.
Thank you.
Thank you.
So Joris, you're the next on the line.
Thank you very much, Aap and Jean-Christophe. I would like to answer to the question of Odile, an excellent question. It's not only excellent, but it's only a complicated question, because I think that after we have seen today, and also from my side, I like to talk the whole consortium for this really excellent and outstanding collaboration. We really worked as a team. That being said, to go specifically to the regulatory on a global basis, it's an extremely important question posed by Odile Leroy. So I think we have to consider we are here, and ZAPI shows that very clearly, at the eve of a paradigm change. So, and when you are at the paradigm change, also from a regulatory point of view, not only scientifically, it asks a lot of intellectual effort in order to go to a kindly different world, also concerning the regulatory for vaccines, because it's not only the platform master file that is showed here. It's also much more, as we have seen, the vaccine by design. It has regulatory impacts. It's also the quality assurance approach, which is totally in vitro. So we totally comply with the 3 R's, which for different reasons starts to be at least of interest. I cannot say accepted, unfortunately, on a global basis. So that being said, of course, we are in contact with WHO, and even in March, you have seen the ICMRA document between EMA, FDA, and 16 other regulatory agencies, which in fact already applied what we have being done at ZAPI. Thank you. Over.
Thank you, Joris. Perhaps before we, I see Bart Haagmans and Jeroen Kortegaard. Before we go into that, I see there's a remark by Ivo Klaassen about the platform master file. Ivo, can I give you the floor first?
Yeah, thank you, Aap, for that. No, I will be in the regulatory session tomorrow, and also in the roundtable discussion closing this meeting. So thank you for a good meeting. I really enjoyed today. But I was asking for the floor to dampen the enthusiasm a bit about already an adoption of the platform master file by European Medicines Agency. We are certainly working on that. We've just put out a concept paper on the development of a guideline on data requirements for the vaccine platform technology master files, veterinary only, I have to admit. I will address that tomorrow as well. It's out now for a consultation period, so I would like to invite everyone to comment on that, because certainly things are there that we can improve. Then after that, we will start with the drafting of the actual guideline, and it's all in relation to the new veterinary regulation that will go into application as of January 2022. But I'm sure that the ZAPI program and of course the ongoing pandemic have accelerated the thinking, also the regulatory thinking, enormously on platform technology. So I'm very convinced that we will see huge developments there in the regulatory area as well. So thanks for giving me the floor now, and just to give you this warning, not to accept too much at this particular moment in time.
Well, we expected you to say that tomorrow as well. So you will come back to that tomorrow, definitely at the end of the meeting. So so I think there has been a courageous effort of yours and his group, you know, to at least to push this area, and I think it's of extreme importance. On the one hand, yeah, we of course there's a lot of people coming with a veterinary background, and that helps a lot, I think, in this arena. But okay, let's take up that point tomorrow. That will give plenty of discussion tomorrow. But you were in line, Bart Haafmans.
It was coming back to the particular presentation of the antigens and the benefit we have seen in enhancing the antibody responses. That's on one hand a quantitative effect, but we haven't really fully explored the qualitative effects, although we have seen also in the RBD experiment with MERS that not only the antibody levels are enhanced, but also the quality, that is the affinity, is quite different in the ones that are immunized with particles compared to the RBD only. So that's one thing. And I think recent studies also hint to the benefit of the particle presentation for the glycosylation of the antibodies and the involvement there. So I think there's still studies needed there to see what also at the level of quality there would be differences that can be used in combination, maybe also with different adjuvants.
Okay, thank you. I think that Jeroen, you still had a question?
No, I was just helping out Ivo, actually.
He doesn't need help for that, I think.
No, he had problems raising his hand, but it was a technical thing. So.
Oh, sorry. Yeah. Okay, but he found out eventually. Yeah, there is a remark here by Berend Jan, and that's about the messenger RNA, the success of the messenger RNA vaccines. Perhaps Berend, could you explain what you mean by that?
Yes, I can. I think the data from the ZAPI consortium show that you need, and as you also indicated, that you need multimerization of the antigens. And we have this nice super glue system, which is, I think, very nice and works beautifully. But if you would like to use mRNA vaccines, then you have to do the coupling already on forehand, so to say. So you have to do molecular coupling and express the whole vaccine antigen from one open reading frame. So I'm wondering how we could achieve that. Whether there are ways of linking the antigens to scaffolding proteins directly, genetically, so to say. And maybe people have thoughts on that.
Yeah, I think that's an obvious thing that has been discussed a long time, and I think the whole thing boils down to antigenic presentation. Yeah? And I think also there's quite a few questions at the moment about adjuvants as well, multimeric presentation, and for myself, I realized that I started to work on that more than 40 years ago, and when we started with ISCOM matrix, and all those kind of things, and I realized the last experiment I did was with the good old alum, you know. So it's a little bit of a little bit of a denial of the enormous advantages have been made. One of the problems we are facing, of course, especially when we're thinking about phase 1 trials that we want to do, that there is only a few adjuvants that have been approved for human use. And if you come with a new technology, like for instance the C1 technology, and in the meantime you come with a different adjuvant system, then you have two variables that makes the process even more difficult. You know? So alum we all know, and if you come, for instance, with a C1 produced antigen, well purified, and it's first in man, you don't want to go first in man with two different new things. Perhaps Joris, you could comment on that.
Well, it's I I would say it's in fact a simple principle. If you have to prove what you say, and I think the COVID situation very clearly shows that the regulators of the whole world, and I would say particularly in Europe, and Emma and the colleagues who are under a lot of stress, they work very pragmatically. They look at the file, they look at the data, and if you have adjuvant, non-adjuvant, multiple adjuvants or not, and we have the same discussion on C1 and on everything what has not been done before. And it's a little bit true as well for the RNA, messenger RNA vaccines. We see that very pragmatically, based on data, they get through and they get accepted. And I think this is a logic which should apply very regularly. And from there, I think everything reasonable is.
So what I learned early in my career, so to say, was never come with two changes at the same moment. That is, you don't agree with that?
Yeah.
With Carmen, you want to comment on that?
Yes, I really will support that. For this is a basic scientific approach, only change one variable at the time. And as we here dealing with veterinary vaccines for food producing animals, we have these nasty things that each new adjuvant has first to undergo the whole MRL assessment, which is time consuming and costly as well. And if we really want to have vaccines for emergency approaches, then we should really rely on these adjuvants which are already approved.
Yeah, Carmen, we have a practical problem there. Yeah, so we have developed together with the Swedish group the matrix M. Yeah, so and which is accepted now. But if we want to use it, we can't get it anymore. So that's a problem. And the same holds true for MF59 and ASO3. It's very difficult because you're going to be in competition then with the production line or the development lines of the companies that bought the adjuvants. If you don't sell it to the companies, then the problem is that it will never be used. So what is the solution there?
Yeah, stay with the old things, even if you do not like them. I have exactly the same feeling. If we have these modern vaccines and take old-fashioned adjuvants, sometimes it looks a little bit difficult. But we have to decide if we have a choice or not. And as long as we have no choice, we have to live what we haven't had.
May I react?
Perhaps before we go on, Carmen, you were quite adamant about never change two things at the same time. Don't introduce two variables. You will be in trouble.
This is solid scientific work. Huh? Only change one variable at a time.
Okay, Joris.
Well, I want to say only one thing. I do not agree with the principle, since that has not been done before, or since we did not use it before, it will be very difficult. I think we have to be pragmatic there. I agree scientifically, don't change more than one thing at a time. But anyway, if you come with solid data and present it, I think there is a more open mind now to look at those things, particularly in the face of the pandemic. At least I hope.
It will take extremely long time, of course. If you come with a new adjuvant that has not been approved yet, and with a new antigen, I think from a regulatory point of view, it's a nightmare, isn't it?
I find it not, but unfortunately.
Yeah, okay, I agree. Ronan, you want to chip in there?
Yeah, I want to say that I agree with this principle, especially if we had a real data that show for C1 antigen that there is a real advantage of using one of those new type of adjuvant, maybe we should consider it. But currently we have good results with the adjuvant that we have used, the alum. So there is no really data that support any changes for that. And coming to the fact, as Carmen said, that we don't want to have two kind of variants, I think altogether at the moment, I would put it in a very low priority to use the new adjuvant in this case.
Okay, so because these are very practical questions, and Carmen was talking about veterinary vaccines, and I'm quite sensitive to that, but also there's questions now, are you really going to use the C1 produced RBD or other constructs in a phase 1 in humans, and then it has to be decided if yes, after the animal experiment, what kind of adjuvant are you going to use? And I think the most pragmatic way of doing it, if it works, it works, or skip the adjuvant altogether and go with a higher dose of antigen. But I think Martin has the solution.
Can I
No, I think the solution was presented or is now used, because if you look at the messenger RNA, if we would have used this rule, not change one thing, it would be never used, because there's 5 new lipids in it. There's messenger RNA in it, which was only tested in a few hundreds of people for cancer. And now it will be in billions of people. So I think this changed, for me, the whole story. Because if I would have tested
Peter,
wait, Peter nails it. I think Peter, he's the expert at the moment. So Peter, help us.
I fully agree with Martin. I think we learned quite some lessons over the last 10 years at the human side. We need to know why you need an adjuvant, but if you need a new adjuvant and you can show why, then people will accept it. And that's what happened with the messenger RNA vaccines.
Exactly.
They're completely new. We see today that the public and Facebook and all these very ugly people that don't know anything about science are against it. But we see the data. We see that it is 95% helping us to protect against the virus. And it is completely new. Did we change one thing? No, we changed everything. So I think we have to be pragmatic, open-minded, and look at the data. And that is what is happening today. Over.
Okay, thank you. Martin, you were waving your hand, and then Carmen.
That was from last time. I fully agree. I think this is now changing, and we should be open also to change more than one part of a vaccine.
I agree with that.
Yes, you are right. And we have to sort out 2 different things. What happens here with the mRNA vaccines is completely new, but it's for humans. And for humans, there is no food producing, humans are no food producing animals, even if some cannibals are around. Okay, but if we use a new adjuvant for food producing animals, then this adjuvant has to undergo the minimal residual limit investigation to make the food of the vaccinated, raised from the vaccinated animal safe for consumers. That means if we, out of the sudden, for a zoonotic situation, use a new technique plus a new adjuvant, this will block the quick licensing. For first, the adjuvant has to undergo the MRL procedure, and this is time consuming. This is what I try to explain. Not again to have new adjuvants, but they have to be checked for their ability to be used in food producing animals. Then they are useful.
That's clear.
Thank you, Carmen. Thank you for that. Perhaps I could give the floor to Jeff. He didn't raise his hands, but I consider him to be a wise man who can advise us there. Go ahead.
You have to ask which question are you addressing to me, Abe? Is it about the speed with which we went with RNA, the future of RNA, or a regulatory question?
It's a regulatory question about, are you going to, if you have, for instance, a C1 produced a spike or a C1 produced receptor binding domain, and you want to use an adjuvant there, you need to use an adjuvant. Are you going for alum or are you going more sophisticatedly? And I think the regulatory experts do not fully agree there. Could you give your opinion there?
I think it depends what that adjuvant is. If it's a new adjuvant, as you said, then there is a lot of work to be done on the safety of the adjuvant. If it's an adjuvant that has been used previously, it's easier, but you still want to make sure that the combination is safe, because sometimes, and I'm afraid it's more often than you would imagine, you've got contaminating host proteins in your vaccine. And when you adjuvant, you run the risk that you induce an immune response against the host proteins, which even if it's then made in a CHO, a hamster or whatever line, you may have a significant homology with the human. And so you've always got that check, that autoimmunity type worry if your materials are not safe and sorry, not pure. And if you adjuvant, your risks of rare autoimmune complications are, I think, more of concern than if you're not adjuvanted. We had a policy in Sanofi, which was that, when we were developing our high dose flu for the elderly, GSK was going with their adjuvanted flu. And we had a long discussion. And we decided simply to put 4 times as much hemagglutinin in there, but no adjuvant. And I remember saying, well, this is madness, because it's pennies anyway. We're putting 4 times as much in, and we can't even charge any more for the product. And Dave Williams at that time said, well, how much do you think it costs for a new adjuvant, right? And in fact, it was cheaper to put more in, but it was also safer, and it won the race. And GSK dropped their product, and we managed to get ours to the market. But our chief medical officer was very clear. She said, Jeffrey, I do not want you to use adjuvants in vaccines that get delivered every year. They are powerful immunostimulants. You can use them in a vaccine that you're going to get once or twice in your lifetime, but not every year, please. And that was her position.
Okay, Jeff, one of the important points you mentioned is that this autoimmunity thing, but I think when we are talking about, for instance, Bachelor or C1, that's a non-issue, isn't it?
Yeah, probably. I mean, there are, probably it's a non-issue, but again, you've still got to be a bit careful with adjuvants.
Okay.
Yeah, it is a new development if there's an adjuvant there, I'm afraid. It's new safety tests, new trials, right?
It causes a lot of extra hurdles.
Thank you, Jeff.
Yeah.
Just to remind you that we are now over time.
I was just going to ask you, because it becomes really interesting now. So
it is quite interesting, but we'll have time also tomorrow to have a lot of discussions.
Yeah, so
I think we need to stop there. We'll answer the questions in written. There are a number of questions on the particles, on slow release, and so on, and all of interest. And we can discuss that tomorrow. I think it's time to close for the day.
Okay. It was a long day. And
I think long day, very, very intense, very active. We did not discuss a lot about how the team and the different partners worked together during these past 5 years. And actually, we exchanged a lot of ideas, materials, and you have seen the flow of information and materials between the different teams. And just to give an example, today it went very smoothly, and we did not rehearse at all because we did not have the time, and the presentations were all very good and perfect. So thank you very much, and see you tomorrow.
Yeah, thank you very much as well.
Thank you to Adi and Mehdi, who are
the organizing team.
In the back
very
office.
Good. Thank you.
Yeah. Thank you. Have a good evening.
See you tomorrow.
Thank you. Bye-bye.
Goodbye.
Bye-bye.
Bye-bye.